Hello guys. Welcome to another video from Ask Me Biotech. In today's video we'll be exploring about what CRISPR-Cas9 is, and how it is used in modern-day genome editing techniques. Also we'll be looking at the steps involved in CRISPR-Cas9 genome editing and subsequent validation of results. Now before we start, I'd like to clarify that this video is an introductory one to the techniques of gene editing, and is intended for the general audience who are enthusiastic in knowing about biotechnology, molecular biology, and CRISPR-Cas9. So let's begin. Let's start with what is CRISPR. CRISPR stands for clustered regularly interspaced short palindromic repeats. It is a sequence of DNA found in bacteria and archaeas. For the ones who do not know what archaeas are a different lineage of microorganisms, who are also known as extremophiles, as they live and thrive in places with extreme conditions, such as high salt concentrations, high temperature, etc. Now the question is, from where do these microorganisms get the CRISPR sequence from? CRISPR sequence is obtained from viruses called as bacteriophages. Bacteriophages cause infection in bacteria. During the course of infection, bacteriophages inject their genetic material into the bacterial cell, which then undergoes fusion with the bacteria's own DNA. But sometimes, these bacteria store a particular sequence from these bacteriophages that enables them to have a memory of previous infection. This is then used to prevent further bacteriophagic invasion. So we now know what CRISPR is. Let's look at another component, that is, Cas9. Cas9 is a protein enzyme that stands for CRISPR-associated protein 9, and belongs to the class of endonucleases. Endonucleases are enzymes that are able to cut DNA at specific sequences within the DNA, hence also sometimes referred to as molecular scissors. Cas9 doesn't cut the DNA sequence at any random site, rather, only at the places where the sequences are complementary to the CRISPR sequence. Cas9 system was discovered as an immunity-providing mechanism in the microorganism Streptococcus pyogenes. It was a significant discovery in the field of gene editing and molecular biology, and its founders, Jennifer Doudna and Emmanuel Charpentier, were the recipients of the Nobel Prize in Chemistry in 2020. We saw that Cas9 was referred to as dual RNA guided. Let's understand these terms. RNA stands for ribonucleic acid. It is the genetic material for many viruses, and is usually single-stranded. It contains uracil instead of thymine in its structure. Dual RNA guided means that the sgRNA, as discussed later, contains two parts, cirRNA and tracer RNA. We are now on the threshold of beginning our journey into the world of gene editing. Now before we start, I'd like to ask that, why do you think CRISPR-Cas9 gene editing is of so much importance to mankind? The answer is, CRISPR-Cas9 genetic engineering technique can be implemented on living organisms. That means we can modify plants to make them produce better yield, provide them with resistance to pests and pathogens, make them more resilient and tolerant to environmental conditions and stresses. It can also be implemented on animals to treat various genetic disorders. Not just that, but gene knockout studies can be performed now that would provide better insight on which genes control what. This is popularly known as reverse genetics. Reverse genetics finds its use in pharmaceutical industries as a tool for target identification and validation. Also, genome can be cut at desired location, with extremely high precision and ease. And as already discussed, this technique was developed by Jennifer Doudna and Emmanuel Charpentier in 2012. Let's look at the stuffs we need to perform a successful CRISPR-Cas9 gene editing. We need cirRNA or CRISPR RNA, tracer RNA or transactivating CRISPR RNA, sgRNA or single-guided RNA, Cas9, of course, and a repair template. Step 1. Selecting an organism. Gene editing cannot be performed on all organisms. So we need to choose which organism we want to work upon with. A good model organism is often mice. Step 2. Selecting target gene. Next step is to select a gene and analyze it using computational studies and bioinformatic analysis. Given below are some of the characteristics of our DNA sequence that we analyze it for. Step 3. Selecting a Cas9 system. All Cas9 systems cannot serve to all purposes. Depending on what type of operation we intend to perform on our gene, we need to choose a suitable Cas9 system. Different companies have different names. Here's some common names of Cas9 systems. Step 4. Designing the guide RNA. The designing of sgRNA needs to be done by computational methods. It has two parts. The crRNA, which is 20 nucleotides long, and is complementary to the target sequence, and a tracer RNA, that identifies the Cas9. Once the Cas9 is identified, the tracer RNA then guides the nucleus to the site of cleavage. The sgRNA thus designed, now needs to be cloned into a plasmid. Step 5. Making a clone of our sgRNA. We now have state-of-the-art facilities to synthesize the oligos of the gRNA, or sgRNA, in vitro. But we have to prepare a stock or clone of it. To do so, select the specific plasmid, insert the gRNA gene in it and develop several clones of it. Step 6. Delivering the sgRNA. The electroporation method is widely used to insert the Cas9 and sgRNA into the target cell. Here, under the influence of the current, pores are created in a cell that allows the nuclease and sgRNA intake. Viral vectors like adenovirus, adeno-associated virus, lentivirus, and retrovirus are used to perform the same function. Finally, our sgRNA is inside the cell. Now comes the time to validate our experiment. 3 common techniques are polymerase chain reaction, in vitro transcription, or DNA sequencing. DNA sequencing is performed prior to the experiment, and after the completion of the experiment, and is then compared. It lets us understand whether a gene is successfully removed or not. PCR validates the experiment by amplifying the target from the modified cells. Step 7. Resealing the knockout. It is often required to introduce point mutations in the original genetic sequence for the purpose of research and observations. For this purpose, scientists use the cell's own genetic repair mechanism, that is, homology-directed repair, to introduce point mutations. That was all about the basics of CRISPR-Cas9 gene editing technique. If you liked the video, don't forget to like, share and subscribe. Thanks for watching.